Bismillah ar-Rahman ar-Rahim. Today we are going to finish the cartilage. We will discuss the matrix of the cartilage. We will discuss the two other types of cartilage, which are elastic cartilage and white fibrocartilage. We will compare between different types of cartilage. We will also discuss the growth of cartilage and nutrition of cartilage. Cartilage is a type of connective tissue, so the extracellular matrix will be formed of fibers and ground substance. The ground substance is formed of macromolecules and tissue fluid. The extracellular matrix of hyaline cartilage appeared as homogeneous and semi-translucent that give it a glassy appearance. This is the homogeneous matrix of the cartilage. The type of collagen in the cartilage is collagen type 2. It is formed of a fine network of collagen type 2. This collagen type 2 does not appear in the microscopic under the microscope because these fibers or these collagen fibers do not form bundles. They form fine network of small fibrils that are submicroscopic, so they do not appear individually in the under the microscope. They also have the same refractive index of the matrix, so they cannot be differentiated under the microscope. Regarding the tissue fluid in the ground substance, they are formed of 75% of the matrix formed of tissue fluid. This high amount of tissue fluid allows easy diffusion of oxygen and nutrients from the perichondrium to the avascular cartilage. The big amount also of tissue fluids makes the cartilage act as shock absorber. And it is important to know that the cartilage is avascular, does not contain blood vessels. Regarding macromolecules, we know from the chapter of connective tissue that it is formed of three main parts: proteoglycans, glycosaminoglycans, and glycoproteins. The proteoglycans are formed of core of protein that have glycosaminoglycans attached to it. This proteoglycans gives the cartilage its rigidity. The glycosaminoglycans either sulfated, like chondroitin sulfate or keratin sulfate, or they may be unsulfated, like hyaluronic acids. The glycoproteins includes adhesive chondronectin. Staining of the matrix of the cartilage. Because the cartilage contains chondroitin sulfate, so they appear basophilic by hematoxin and eosin. This chondroitin sulfate mainly accumulate around the chondrocyte lacuna, gives it more pisophilia, and we call it territorial matrix. So the matrix around the chondrocyte lacuna appears more pisophilic than the other matrix due to condensation of chondroitin sulfate. Also, the cartilage can be stained by periodic acid sheaves because it contains glycoproteins. It can be stained metachromatically due to its content of glycosaminoglycans. This is the second type of cartilage, which is elastic cartilage. In fresh state, they appear yellow due to presence of elastic fibers, which gives it its elasticity. This type of cartilage is more pliable and distensible compared to hyaline cartilage. Regarding its size, it is present in sites where flexibility and support are needed, like the ear pinna, like the external auditory canal of the ear, and the eustachian tube, and the epiglottis. These sites all begin with E, are the sites of elastic cartilage. All these sites needs flexibility, so they contains a type of cartilage that is rich in elastic fibers. The structure of elastic cartilage is similar to hyaline cartilage, except the matrix contains network of elastic fibers. That's why it appears yellow in fresh state, because elastic fibers appear yellow in fresh states. These elastic fibers condensate around the chondrocyte lacuna. As we see, these fibers can be visible under the microscope, in contrast to collagen type 2, the ordinary collagen type 2 of the cartilage. These fibers, elastic fibers, can be seen under the microscope. Also, chondrocytes are present singly or in 2 cells inside the lacuna. The third type of cartilage is the white fibrocartilage. In fresh state, they are seen white in color. They are present in sites that subjected to severe stress and weight bearing, like the intervertebral disc, the symphysis pubis, the semilunar cartilage of the knee, and the site of insertion between the tendon and the bone. We know that the muscle is inserted in the bone by tendon. The site of insertion of tendon, or the junction between tendon and bone, is formed of white fibrocartilage. Regarding the structure of white fibrocartilage, it is formed mainly of collagen type 1, parallel bundles of collagen type 1 containing blood vessels. This is the collagen type 1. Between these collagen type 1, we have rows of chondrocytes surrounded by small amount of cartilage and extracellular matrix. So the component of white fibrocartilage are collagen type 1. This collagen type 1 contains blood vessels. Between these fibers, we have rows of chondrocytes with small amount of extracellular matrix. It is very important to know that the white fibrocartilage is not covered with perichondrium. One of the important sites of white fibrocartilage is the intervertebral disc. The intervertebral disc is formed of central gelatinous material called nucleus pulposus that is surrounded by a ring of white fibrocartilage called annulus fibrosus. With aging individual, this is the normal disc formed of gelatinous material nucleus pulposus surrounded by the white fibrocartilage. With advanced age, this fibrocartilage becomes thinned and weakened, which leads to protrusion of the gelatinous material that leads to compression of the spinal nerve. This compression of the spinal nerves causing severe pain that leads what is known as a slip disc or disc prolapse. Regarding growth of cartilage, we said that the chondroplasts are present in the inner chondrogenic layer of the perichondrium, and it is responsible for the first type of growth, which is called appositional growth of cartilage. Appositional growth of cartilage means addition of new cartilage at the surface of existing cartilage. It also means addition of cartilage under the perichondrium. This is the function of chondroplasts. The second type of growth occurs by interstitial growth, which occurs by chondrocytes. We said that chondrocytes divide by mitosis and gives different chondrocytes either inside the same lacuna, we call it cell nest, or 4 to 8 cells in separate lacuna. This leads to expansion of cartilage from within, plus its ability to form the extracellular matrix. So the chondrocytes are responsible for interstitial growth of cartilage, the chondroplast appositional growth, and the chondrocytes interstitial growth. We said in the last lecture that the articular cartilage is not covered by perichondrium. So the type of growth in the articular cartilage is only by interstitial growth because it contains only chondrocytes. It does not contain chondroplasts. Regarding nutrition of cartilage, we said that cartilage is avascular connective tissue, so it takes its blood supply from the blood vessels that are present in the outer fibrous layer of the perichondrium. Blood vessels are present in the outer fibrous layer, carry nutrients and oxygen to cells of the cartilage that diffuse through the cartilage matrix and takes the waste products back to the blood. We said in the last lecture that we have two types of cartilage that does not contain perichondrium, which are the articular cartilage, this hyaline cartilage does not covered with perichondrium, and the white fibrocartilage also is not covered with perichondrium. So nutrition of articular cartilage occurs via synovial fluid and blood vessels present in the synovial membrane. Nutrition of the white fibrocartilage occurs by blood vessels that are present in the collagen fibers, collagen type 1, that form the main structure of the white fibrocartilage. We have to know that the hyaline cartilage is susceptible to degeneration and calcification with aging. But because this cartilage is avascular, so healing and regeneration of damaged cartilage is always slow and is not complete. To compare between different types of cartilage, appearance in fresh state of hyaline cartilage appears as glassy or bluish translucence. White fibrocartilage appear white, and elastic cartilage appear yellow in color. The site of hyaline cartilage, it presents in costal cartilage, in the fetal skeleton, in some respiratory cartilage as larynx, trachea, and bronchi, and in the articular cartilage and epiphyseal plate. The white fibrocartilage is present inside with severe stress and weight bearing, like intervertebral disc and symphysis pubis, while elastic cartilage is present in sites where flexibility is needed, as ear pinna, epiglottis, Eustachian tube. The perichondrium is present in all types of hyaline cartilage except the articular cartilage. The perichondrium is absent in white fibrocartilage, and it presents on all sides of elastic cartilage. Regarding the abundance of the cartilage, the most abundant type of cartilage is the hyaline cartilage. This is an important question, the types of fibers in the extracellular matrix of cartilage. In hyaline cartilage, we have the ordinary type of collagen, which is collagen type 2. The white fibrocartilage contains collagen type 2, as it is a type of cartilage. It also contains collagen type 1 that forms the collagen bundles that run between the chondrocytes. Elastic cartilage contains collagen type 2 as the ordinary collagen. It also contains elastic fibers in the extracellular matrix. Regarding the abundance of extracellular matrix, the most abundant matrix is present in the hyaline cartilage, while in white fibrocartilage we have a minimal amount of extracellular matrix between rows of chondrocytes. The strength of that cartilage, the most strong or the strongest cartilage is the white fibrocartilage. That's why they are present in sites of severe stress. The cells in different types of cartilage, the hyaline cartilage contains both types of cells, the chondroplasts in the perichondrium and chondrocytes in the matrix. The white fibrocartilage contains only chondrocytes. They do not contain chondroplasts, as they lack perichondrium. The elastic cartilage also contains two types of cells, chondroplasts and chondrocytes. To summarize what we have taken, we must know the components and staining of cartilage matrix, types of collagen in different types of cartilage. The hyaline cartilage contains type 2, white fibrocartilage contains type 2 and type 1. The elastic cartilage contains type 2 and elastic fibers. We have to know the site and the structure of elastic cartilage and white fibrocartilage. Nutrition of cartilage occurs via blood vessels in the perichondrium. In types of cartilage that lack perichondrium, blood vessels, nutrition of cartilage in articular cartilage occurs in the synovial fluid, and in white fibrocartilage via blood vessels in the collagen type 1. Growth of cartilage occurs by two types, either appositional growth that occurs by chondroplast or interstitial growth occurs by chondrocyte. Thank you.